Hi everyone. Today we are going to see an important topic from immunology subject, T cell receptor. T cell, T means thymus derived. It originates in bone marrow and migrate to thymus. The maturation in thymus to become T lymphocytes. And then it migrates to bloodstream. It encounters the antigens and recognizes those antigens by T cell receptor, which is further activate the adaptive immune response. In this lecture, we study TCR and CD3, that is cluster differentiation 3, which is a signaling protein associated with TCR, T cell receptor. The TCR is a protein complex found on the surface of T cells, membrane spanning proteins. T cell receptor interact with MHC, major histocompatibility complex, on the surface of antigen presenting cells or target cells. Each T cell carries between 10,000 to 30,000 identical copies of a single TCR. Types of TCR. Two types of TCR heterodimers present: TCR alpha beta 95% and TCR gamma delta 5%. T cell express either alpha beta or gamma delta receptor but not both. First one, TCR alpha beta type. It composed of two polypeptides. Alpha chain is about 49 kilodaltons and beta chain is about 43 kilodaltons. Each chain has two external extracellular domains. Outermost domain is known as variable domain B and the other domain is known as constant domain C. Each of the domain is stabilized by the internal disulfide bonds. The variable domain of the both chain form the antigen binding sites. In this diagram, we can see both variable and constant domains. After TCR recognize the antigen presented by an MHC molecule, a signal is transmitted from the surface of the T cell to the nucleus, which leads to T cell activation. But TCR cannot do this alone because cytoplasmic tail of the TCR is extremely short. For this, T cell receptor are associated non-covalently with a complex of proteins. The complex of proteins is made up of four different types of proteins: gamma, delta, epsilon and eta. Two heterodimers formed by these proteins are known as CD3, cluster of differentiation 3. First heterodimer is delta eta and second heterodimer is gamma epsilon. Two eta chains linked together by the disulfide bonds forms the remaining part of this protein complex. The cluster of differentiation 3 proteins and eta chains are identical in all T cells. So, T cell receptor is a complex of proteins. The alpha and beta proteins of this complex do the recognition of antigen. They cannot signal. For signaling job, gamma, delta, epsilon and eta proteins are there. They initiate intracellular signaling pathway after antigen recognition by T cell receptor. This picture shows how the antigen presenting cells is representing and how the TCR is getting attached to the antigen and how it is getting activated after receiving the antigen signal from the target cell. So this video we have seen the basic structure of T cell receptor and its signaling. This picture explains the comparison between B cell receptor and T cell receptor. So next video we will see some important topic from immunology. Thank you.